Hi everyone, welcome to the second webinar in our 6-part series on alternative approaches for acute inhalation toxicity to address global regulatory and non-regulatory data requirements. My name is Amy Klippinger. I am the Associate Director of the PETA International Science Consortium and will be co-moderating today's webinar with Dr. David Allen, who works as a contractor supporting the USNTP Interagency Center for the Evaluation of Alternative Toxicological Methods, also known as NICETA. I'd like to thank you all for joining this webinar, and before I introduce today's speakers, we'll cover a few logistics. This is the second webinar in our 6-part series. The first webinar focused on current practices for acute inhalation toxicity testing. And if you missed that webinar, I highly encourage you to watch the recording, which is posted online at the link on your screen. Today's webinar and the other remaining webinars will look past the current practices towards the development and implementation of alternative approaches that reduce and replace acute inhalation testing in animals. The slides from today's webinar and a recording will be posted within a couple days, again at the link on the screen. Also, after today's webinar, there should be a feedback form that pops up in your internet browser. Please fill that in if you have time. Everyone is muted now. If you have a technical question for me anytime during the presentations, you can use the question box, which should be located in the panel on the right-hand side of your screen. If you have a question for one of the speakers, you can type it in that same question box. We will hold all the questions until after both talks, and as time permits, David will ask your questions to the speakers during the Q&A period, so please do send them in anytime during the webinar. And lastly, before I introduce our first speaker, I want to mention the other webinars coming up in the series. The next webinar on in silico methods will take place on May 26th. You can find more details about this and the other upcoming webinars online. Please note that if you're registered for this webinar, you'll automatically be registered for all 6 webinars in the series. Speaking first today will be Dr. Mariana Gata. Mariana leads the preclinical assessment team in the British American Tobacco R&D Center in the UK, applying in vitro approaches to the assessment of current and future tobacco and nicotine products. Since joining British American Tobacco, Mariana has had a number of roles supporting the science to help characterize the biological effects of tobacco products, including the development of in vitro models of tobacco-related diseases, such as chronic obstructive pulmonary disease and cardiovascular disease, and the development and application of in vitro smoke systems. The title of her talk is Acute Inhalation Toxicity, In Vitro and Ex Vivo Systems.
Thank you for the introduction, Amy, and good afternoon and hello to all the participants of this webinar. Thank you to PETA International Science Consortium Limited and to NICETA for organizing this very interesting webinar series on alternative approaches for acute inhalation toxicity and for inviting me to present. I have been asked today to discuss the fundamentals associated with in vitro and ex vivo inhalation toxicity systems and their potential for application in non-animal testing approaches. So the approach I'm going to take today will be to give a brief introduction into the lung, the anatomy and the deposition, and what can be and is modeled in vitro to provide an in vitro toolbox, followed by a number of case studies or examples outlining our initiatives in developing appropriate in vitro strategies for product assessment. I will briefly touch on the diversity of our products and how we generate test materials, and then final up with a summary. So the role of the respiratory tract. The respiratory tract is one of the major routes of human exposure and is the interface between the environment and the internal organs. In its basic form, the respiratory tract is made up of 2 lungs and a series of air passages leading to and from the lungs, also known as the conducting airways. The extrathoracic or upper portion is made up of the nasal and oral cavities, pharynx and larynx. The bronchial and bronchiolar, or lower portion, consists of the trachea, which branches to give rise to two primary main bronchi, one into each lung. Within the lung, they further branch into progressively smaller airways called bronchioles, the smallest of which are called terminal bronchioles. Within the respiratory alveolar portion, the terminal bronchioles give rise to respiratory bronchioles, which ultimately lead to the alveoli, the small balloon-like sacs. To give an indication of the surface area of the lung for gaseous exchange, if the 700 million or more alveoli found in both lungs are to be flattened out to the size of a tennis court, it is remarkable to think that this is all neatly folded and bundled into the chest cavity. Yes, hello?
Can you hear me? This is
yes,
Dave. You're fading in and out just a tad bit. So if you could, I don't know if you need to move closer to your machine or what.
Okay, can you hear me now?
That's much better.
Is that better?
Thank you.
Okay.
Yeah.
All right then. Excellent. All right then. Right, so moving on from the respiratory tract region. So as the air passes through the respiratory tract, it becomes conditioned or prepared for delivery to the lungs. It is warmed and moistened and freed of particulate matter, which is a function of the cells lining the tract. So what cells are found in the respiratory tract? In the tracheal bronchial region, this is lined with pseudostratified epithelium containing three common cell types: the ciliated cells, goblet cells, and basal cells. The cilia are small hair-like cellular projections that extend from the surface of the cells. The goblet cells are specialized epithelial cells that secrete mucus to coat the lining of the respiratory tract and are interspersed among the ciliated cells. The cilia move together to push mucus secreted by the goblet cells away from the lungs. Particles of dust and even pathogens like viruses, bacteria, and fungi in the air entering the lungs will stick to the mucus and are carried out of the respiratory tract. In this way, mucus helps to keep the lungs clean and free of disease. Further down in the respiratory bronchioles, these are narrow channels less than 2 millimeters in diameter, and they are lined by a simple cuboidal epithelium consisting of ciliated cells and non-ciliated clara, surfactant-secreting cells, also known as club cells, and these are unique to the bronchioles. Further down, the alveoli have smaller thin walls to aid diffusion and maximum absorption of oxygen into the bloodstream and consist of alveolar type 1 epithelial cells and type 2 epithelial cells. The type 2 function to make the surfactant to help gaseous exchange and offer a stem cell-like capacity to repair the damaged alveoli. So what happens during inhalation? Where are the particles deposited in the respiratory tract? So the larger airborne particles, greater than 10 microns when inhaled, usually impact on the walls of the nose and the pharynx into the sticky mucosa. A small percentage of particles, less than 10 microns, pass through the nose and the nasal pharynx, upper airways, to enter into the lower airways. Less than 2% of the larger particles reach the lower intrathoracic airways. The great majority of inhaled particles, between 3 and 10 microns in diameter, are deposited on the tracheobronchial surface. Their position at the alveolar level occurs mainly for the particles between 0.5 and 3 microns. Therefore, the conducting airways are very effective in clearing the air of suspended particles, so that the volume of the large particles to be cleared by the alveoli is absolutely minimal. So in developing alternative approaches to inhalation toxicity, there are a number of well-developed and characterized cellular systems to utilize. These cellular systems all have their pros and cons and are useful to consider when developing a tiered or battery approach to in vitro testing. In short, in vitro tests are designed to generate rapid initial data that will give general insight into disease mechanisms and the biological effect of test compounds and materials. And as a result, there's a general shift in employing human cells and tissues to ensure as many of the physiological parameters are maintained in the in vitro test systems. So moving on. This slide highlights the in vitro toolbox offered to inhalation scientists on a sliding scale of physiological relevance, ranging from the very simple and basic monolayer cultures onto the more complex organotypic 3D tissue models and organ-on-a-chip approaches. But let's take some of these in order. So at the top end of the scale are embryonic and induced pluripotent stem cells. This technology and approach offers a promise of facile in vitro studies, but their applications are far too early to be fully recognized in this context. Moving down, we have continuous cell cultures. These are single cell types that can be propagated easily in culture, and majority proliferate indefinitely, either through a natural or introduced mutation. If continuous cell lines have originated from embryos or tumours, such as the H292 cells, or transformed cells, such as the Vs2Bs. These cell lines present advantage of almost limitless availability, easy to culture and handle, and convenient. Cell lines are homogeneous populations, therefore reducing donor-to-donor variability. The disadvantages of continuous cell lines are that they do not retain many features of the original tissue. They may not accurately represent the in vivo situation. The more physiologically relevant are the primary cell cultures that are explanted directly from either a healthy or diseased tissue. The preparation of primary cell cultures is labour-intensive and can be maintained in vitro for only a limited period of time, up to a maximum of 2 to 3 weeks. Primary cells usually retain many of the differentiated characteristics of the cell in vivo, including their metabolic capability and physiological characteristics. The disadvantage is these cells have a limited lifespan and can have a large variation between donor to donor. The 3D systems, however, offer the advantage of culturing cells in a familiar physiological milieu consisting of many cell types and often basement membranes, enabling cell-to-cell and cell-to-matrix connections offered in normal physiology. Scaffolding techniques using extracellular matrix, proteins, peptides, or polymer-based materials can be used to support cellular and tissue structures. Non-scaffold techniques can also be used.
Hey,
yes.
Mariana, I'm so sorry to interrupt you again. We're getting comments that it's hard to hear you. You're kind of going in and out, and I think it would be more clear
on the
if
phone.
you could, yeah, if you were on the phone. Do you have a phone handy?
Yeah, I could try and dial in.
Okay, so it's just on your control panel on the right under audio. You should have the option for either telephone or mic and speakers. So if you click telephone, it will give you the phone numbers. And there should be one for the UK there. Sorry, everyone, just 1 second.
Hello?
Hey, Mariana, we can hear you.
Can you hear me now? Is that better?
Yes, it is. Thanks so much. Sorry, everyone. Thanks for holding on.
That's all right. Apologies, everyone. Must be this, obviously the distance between me and you. Quite far away. Okay, so where do we get to? So we're talking about the 3D systems and how they offer the advantage of culturing cells in a physiological milieu consisting of many cell types and basement membranes. So we talked about the opportunities of using scaffolding techniques, using a variety of different proteins to be able to support the cellular and tissue structures. There are also non-scaffolding techniques that can be used to develop and culture these 3D tissues, such as rotating bioreactors, hanging drops, and magnetic levitation or the promise of 3D printing. Commercially available 3D lung systems offer researchers the ability to purchase robust, fully differentiated lung tissues with quality assurance and reduced development times, which is a huge advantage. However, the disadvantages include the longer time to grow and culture these systems, and these are often more expensive compared to continuous cell cultures, and offer the challenges of donor-to-donor variation. So as we get even closer to an in vivo scenario, we see these organ slices, in particular precision-cut lung slices, and these can be maintained in vitro, having the potential to preserve all cell types present in the original tissue in the correct spatial orientation and having intracellular interactions. These lung slices retain the metabolic activity of the lung parenchyma and can be used in studies requiring bioactivation. However, large variation exists between sequential slices and also from donor to donor. One major disadvantage of lung slices is their limited lifespan, approximately 1 week in vitro. Therefore, their utility is currently restricted in short-term studies. And then finally, we have the lung on a chip and some microtissues. And these offer an even closer bridge to the human scenario, with other key physiological parameters added, such as mimicking the complicated mechanical and biochemical behaviors of a human lung. So inhalation of cigarette smoke, occupational and environmental pollutants are often the main causes of a number of lung diseases, but predominantly chronic obstructive pulmonary disease, or COPD, and this can affect all the major components of the lung. As a result, we are currently developing and applying in vitro models of COPD in assessing the reduced biological activity of our next generation products. To help in designing the appropriate in vitro models, we are using an AOP framework to identify the key events in disease pathogenesis that we can potentially model in vitro. Okay, so moving on to these AOPs. So AOPs are a framework used to organize data and information across a series of linked or causally connected key events. They have a common structure beginning at the molecular initiating event, with intermediate steps leading to an adverse outcome. In this case, the initiating event is tobacco exposure to the lung epithelium, and the adverse outcome are a number of clinical manifestations, being chronic bronchitis, emphysema, small airway disease, for example. The AOP framework can help to identify research gaps and also qualify specific endpoints or biomarkers. In this example, generated by Dr. Holger Bursting of the Institute for Vitro Sciences, some of the key events in the development of COPD have been mapped out. Using this framework, I will show you some examples of how we are developing in vitro models to measure and assess some of these key events, mainly looking at oxidative stress, cellular damage, inflammation, and a number of other endpoints. So, the test article is also an important consideration when developing in vitro test systems. For example, if you take the various products we are testing in our laboratories today, they come in a number of different formats. At one end of the scale, on the left hand of this diagram, we have been accessing conventional burned-down tobacco products containing tobacco leaf that is burnt at a temperature over 800 degrees centigrade. And at the other end of the spectrum, on the right hand side of this figure, there are medically licensed nicotine delivery devices that deliver cold nicotine using puff-activated devices. In between, we have this huge raft of our next generation products that include tobacco heater products, THPs, and e-cigarettes. There are many different types available on the market with differing size, shape, and power outputs, and as shown, have differences in the mouthpiece size, device body size, and weight. Activation of these products vary, and with some being puff activated and others are button activated. And all these different physical parameters need to be taken into consideration when generating aerosols in vitro testing. So how are we generating the test article and exposing cells for biological testing? In general, aerosols from these products are generated using a variety of methods, and the matrix captured in a number of ways. Aerosols are generated from cigarettes and next-generation tobacco and nicotine products are complex, and they can be divided into particle, vapor, and droplet components, depending on the product. There are several methods to track either component or whole aerosols exposures to cell and tissue cultures. The three main types of matrix generated for in vitro tests are particulate matter, aqueous extracts, and direct whole smoke exposures in an aerosol exposure chamber. So the particulate matter. This is a relatively simple, quick, and robust method in capturing particulate matter on a Cambridge filter pad and has been used for many years traditionally for toxicity studies. Solvents used to extract the particulate matter captured on Cambridge filter pads can also affect the way in which cells respond. For example, DMSO, a known solvent, is also a known antioxidant. The TPM can be added to submerged cultures or cells at the air-liquid interface. Aqueous extracts are generated by drawing aerosols through impingers containing either cell culture media or physiological buffers, such as PBS. The aqueous extracts can be diluted and added to cells in submerged culture conditions. Cells can also be exposed to the whole aerosol within a specially designed exposure chamber, which holds the cells at the air-liquid interface, or ALI. This exposure method was developed in response to the challenges of making in vitro exposures akin to the in vivo scenario. Air-liquid interface exposures are more physiologically relevant, where the tissues or the cells are exposed apically to aerosols. This method has many advantages over the previous two methods described, and equally many challenges when generating. With the newer e-cigarette products comes the potential also to assess direct e-liquids in submerged exposure conditions as a quick screen and a proxy for the real matrix. With all of these different matrices that we have explained, there are associated challenges and dose implications which need to be taken into consideration for each exposure scenario. So laboratory study inhalation toxicology either use in-house or commercially available aerosol generators to generate whole aerosols for in vitro studies. In our labs, we use two commercially available systems, the Borgwell Arm20S smoking machine shown on A, and the VitroCell VC10 smoking robot shown in B. These machines vary in design and capability, but in principle their purpose is shared. As a result of many years of development, these generators are very sophisticated and capable of generating aerosols with high levels of repeatability and reproducibility. There are many different types of exposure chambers that can be used in combination with these generation systems and in vitro cultures. With the Borgwell Arm20S engine, we use a chamber that we have designed and developed in-house. The exposure chamber is very simple in design. It is compact, robust, easy to clean, and relatively economical. We have also shared these chambers along with the design blueprints to laboratories developing air-liquid interface cultures. Currently, we know that these are being used across a number of different laboratories in academia, in different industry sectors, and defense establishments. With the VitroCell VC10 smoking robot, we employ the use of the VitroCell linear modules. These have special design features such as individual warm media supply to each of the cell culture inserts used within the chamber. We have extensively researched and characterized these two systems in our laboratories, along with their associated chambers, so that we know and understand the aerosol we are generating at source and at delivery to our in vitro cultures. We're aware of particle size, particle number, deposition, and losses in the system using a variety of dosimetric analysis tools. So on to some case studies showing the utility of in vitro and ex vivo models. All studies presented here, the products tested will include the Kentucky reference tobacco product 3R4S, and in some cases a commercially available e-cigarette. So in this first example, we are using primary human bronchial epithelial cells that have been cultured for over 28 days at the air-liquid interface. During this process, the cells undergo epithelial differentiation to form a mucociliary epithelium, similar to that seen in the tracheobronchial epithelium. During the 28 days of culture, cells are exposed apically to tobacco smoke TPM. Transmission and scanning electron micrographs confirm morphological differentiation. Cells at initial culture formed a simple epithelial layer, while at 28 days, we could see basal, ciliated columnar epithelial cells and mucin-containing goblet cells. After 28 days, the cells are assessed for viability using neutral red uptake and barrier integrity using TERs, or transepithelial electrical resistance. Viability remained constant, but at the higher concentrations, we noted a decrease in TERs. Using two quantitative techniques, flow cytometry and immunohistochemistry, the expression of the goblet cell marker MUC5AC was investigated. TPM treatment caused a concentration-dependent increase in the percentage of MUC5AC positive cells. A slight drop with the highest dose in TPM was noted, but is correlated with a reduction in TERs. So in another example, we are demonstrating the utility of continuous cell lines with exposures to whole aerosol. In the first experiment on the left panel, the study was to investigate the use of single-cell gel electrophoresis, or COMET, to assess DNA damage upon whole aerosol exposure. H292 cells were exposed to different whole smoke concentrations at the air-liquid interface, followed by encapsulation in the low melting point agar suspension, live and subjected to electrophoresis. The fragmented and damaged DNA migrates through the gel and the cell nucleus, from the cell nucleus resembling a comet as shown here. Our studies indicate that as you increase the dose of whole aerosol, the percentage of DNA damage increases in a concentration-dependent manner. In another example on the right-hand side panel, we investigated cytotoxicity and DNA damage potential of e-cigarette versus a reference cigarette. H292 cells were exposed to increased concentrations of e-cigarettes and cigarettes, and cytotoxicity was assessed using neutral red uptake. With the reference cigarette, we elicited a cytotoxic dose-response curve with increasing smoke concentrations. However, with the e-cigarette, these effects were less apparent. We took the same products and assessed the DNA damage using the marker gamma H2AX in these two B cells. In this study, 3R4F was genotoxic, increasing gamma H2AX phosphorylation with increased dose. However, no effects were observed with e-cigarettes. So the next example will focus on the use of commercially available 3D organotypic tissues, namely Mucilaire from Epithelix and Epi-Airway from Matec. Both systems come ready to use and consist of human-derived tracheobronchial epithelial cells that contain ciliated and mucus-producing cells. Both models mimic the in vivo respiratory tract phenotypes, having barrier and mucociliary responses, and can model infection and toxicity responses. These cell types are also amenable to acute or long-term chronic studies, especially the Mucilaire that has a shelf life of up to 1 year. These cellular systems enable morphological and functional multiple endpoint analysis, including production of mucus, ciliary beat frequency, assessment of tight junctions, and so on. So, one example of the functional marker includes measuring the frequency at which the cilia on the epithelial cells beat in response to treatment. These images can be easily captured using digital high-speed image recording, and frequency of cilia beats can be counted either manually using slow-motion playback of a video film, which is very tedious, or incorporation of various software programs and algorithms. The impact of different substances on the rate at which the cilia beats can be observed and compared. So as shown in this short video, looking at the apical surface of the cilia cultures, we can visualize and monitor cilia beat frequency. So in this study, we have investigated the use of epi-airway tissues as a model of respiratory irritancy for tobacco and e-cigarettes. The epi-airway tissues were exposed to either 3R4F whole smoke or aerosols from e-cigarettes, and irritancy assessed using MCT and TER. When the cells were exposed for up to 6 hours, a clear concentration-dependent cytotoxic effect was observed with the 3R4F whole smoke. However, with the e-cigarette, at the 6-hour time period, there was not a response elicited. This example indicates the use of Mucilaire tissues. The aim of this study was to use multi-platform approaches and knowledge-based mapping to compare perturbations in Mucilaire tissue after short repeated exposures to cigarette smoke and e-cigarette aerosols. So on the top left-hand panel, a panel of pro-inflammatory mediators, including 30 cytokines and 3 matrix metalloproteases, were assessed in the media of treated Mucilaire cells and showed distinct responses. 3R4F treatment showed significant increase in a number of cytokines, including VEGF, MMP9, interferon gamma, IL-6, 8, 10, and 4, plus IL-12, 24 hours after the short repeated exposure to 3R4F. And there's little response with the e-cigarette treatment. We looked down to the left-hand bottom panel. We looked at RNA expression profiling, and this was performed in treated Mucilaire cultures using massive parallel RNA sequencing. And we also have looked at this by microRNA expression evaluated by microarray. The number of differentially expressed microRNAs and RNAs, and also alternative splicing events, were greater in the 3R4F exposure conditions compared to the e-cigarette exposures. When we combine data from different platforms with knowledge-based and ontology mapping, we can predict altered and functional links to disease-associated phenotypes. In this case, gene enrichment analysis was conducted on the 3R4F treated Mucilaire cells, and we identified perturbations in oxidative stress response, inflammation, and xenobiotic metabolism response pathways. It has been suggested that 3D lung cultures also make airway surface liquid, ASL, similar to sputum in vivo. In this study, we investigated if the in vitro ASL had good relevance to the human in vivo scenario using proteomic profiling. Untargeted proteomic profiling using LC-MS-MS resulted in the identification of a large number of proteins associated with a number of key biological processes. Further targeted proteomics using multiple reaction monitoring approaches, or MRM, detected 163 targets and proteins in the in vitro ASL. Comparison of the in vitro ASL with clinical sputum samples resulted in a strong overlap of 112 common proteins, suggesting a good similarity between protein profiles from clinical samples and 3D in vitro lung models. So then moving into precision cut lung slices. Lung slices for in vitro cultures have been produced from a variety of species, including rats, hamsters, guinea pigs, and humans, to name a few. In the literature, there are many different methods available that have not been standardized with respect to slice preparation, slice thickness, stabilization, and culture media. In general, the tissue is stabilized by perfusion of agarose through the airways prior to slicing. Tissue cores are then obtained from which precision cut slices are generated, either using tissue slices or mechanical slices. The slice thickness is an important consideration for cell injury, functionality, and tissue integrity. To date, there have been very few studies that have utilized lung slices to model cigarette smoke exposure. Several studies have also used lung slices to examine the effects of cigarette smoke constituents, including acrolein, benzopyrene, and cadmium. In this study, rat lung slices were exposed to 3R4F cigarette smoke for 30 minutes for 3 consecutive days, and viability was assessed using MT2. It was shown that cigarette smoke and vapor phase demonstrated clear concentration-dependent cytotoxic effects, with over 85% of the cells surviving after a 4% smoke exposure. Histological observation also showed that the alveolar structure remained largely intact with no treatment and up to 4% smoke and vapor phase treatment. Higher doses of cigarette smoke resulted in the substantial loss of alveolar epithelium. So, in summary, as we described in this presentation, there are many in vitro tools available to evaluate acute inhalation toxicity, but there are a number of considerations to bear in mind. There are an array of complex systems, and each can be used to address specific aspects of disease processes. However, it is important to note that each tool should be evaluated as a component part of an integrated or weighted evidence approach, and not solely in isolation. It is key to understand what the cells are being exposed to during the studies to help correlate or extrapolate to the in vivo scenario. The consideration of the length of exposure is also key, with some in vitro systems now lending their functionality for longer-term exposures and also repeated dose exposures. To add further reliability to the data and confirm robustness of in vitro systems, there is a need to demonstrate fitness for purpose of the in vitro models and appropriate exposure devices. The AOPs could help to identify gaps and strengthen biomarker discovery, identify key events, and bridge the gap between in vitro and in vivo. So I've described a number of approaches that we're using at BAT. These in vitro and ex vivo systems can be also used in other industries to evaluating the effects of different aerosols and gaseous mixtures, such as pollutants, diesel particulates, and inhaled pharmaceuticals to examine exposure scenarios. And so then finally, I would just like to acknowledge the many people in the laboratories here in Southampton and our collaborators who have supported the development and application of these models. So thank you very much.
Thank you, Mariana. As I mentioned, we will hold all questions until the end, but please send in any questions that you may have via the question box now. So speaking next will be Dr. Annie Jerbec. Annie is the Deputy Director of the Human Health Risk Assessment National Research Program in the Office of Research and Development of the U.S. Environmental Protection Agency, and a senior toxicologist in the immediate office of the National Center for Environmental Assessment of the Research Triangle Park Division. Annie has significant experience and training in inhalation toxicology in both laboratory and clinical environments, dosimetry modeling, risk assessment, and decision analysis. The title of her talk today is What a Difference the Dose Makes, Dosimetry Approaches to Aid Experimental Design, Evidence Integration, and Inferences for Risk Assessment. And Annie, are you on mute? Annie, if you're speaking, we're not able to hear you. I think you're on mute.
Can you hear me now?
There you are. Great.
Sorry about that. Technical difficulties to overcome. Good afternoon or good morning, everyone, depending on where you're located. I, too, want to very much thank both the International Science Consortium of PETA and NICEDM for this opportunity. I very much look forward to talking to you about what's been a passion of mine, which is dosimetry and dosimetry modeling for risk assessment. So my slides aren't advancing.
All right, let me pull them up from my end, and if you just want to let me know when to advance, I can scroll through them.
Next slide.
Okay.
So this is the requisite disclaimer that the views in this presentation are those of myself and do not represent US EPA policy. Next slide. The topics that I will cover today are the conceptual basis, although Dr. Gata did a wonderful job of presenting the human respiratory tract, really setting context for in vitro extrapolation and needing to rely on interspecies extrapolation to do some of the verification. We really need to look at the critical determinants of comparative inhaled dose. I want to set that in the context of where we are at with contemporary toxicology, particularly because dosimetry modeling has matured quite a bit recently. And the context would be multi-scale characterization for risk assessment, particularly to advance the concepts of adverse outcome pathways and mode of action to help evidence integration. And then I will end with a few specific challenges and considerations specific to in vitro testing systems embedded in this larger context of a verification matrix of human and inhalation testing in laboratory animals. Next slide. So the conceptual basis for my talk, I realized that timing is short and we have a lot of material to cover, so I wanted to give 3 take-home messages for this conceptual basis. As illustrated on the right-hand side, we've been working on methods and models to extrapolate laboratory animal data to human context for some time, and I believe, or I'm asserting in this presentation, that those methods and models for interspecies extrapolation can be quite useful to create context for characterizing the in vitro systems and results for better evidence integration. Inhalation dosimetry involves current understanding and a critical comprehension of biological features of the respiratory tract in a comparative fashion, the differences between the species and how they interact with physicochemical properties of the inhaled agent, either particle, fiber, or gas. And you also need to consider the transport and transformation aspects of the exposure generation and the characterization, such as the analytical methods used, as well as how those interact with the physicochemical properties of transport and transformation in that exposure system, be it for an in vivo exposure or for in vitro. Next slide. So again, I'm emphasizing comparative airway anatomy, just a few highlights to follow up on what Dr. Gata already presented. Typically, we break the respiratory tract, particularly for this type of modeling and application into risk assessment, into 3 major areas: the upper airway, upper respiratory tract, the tracheobronchial, and the pulmonary region. The reasons for that being that there are very dramatically different deposition and uptake mechanisms as well as cell types. The dimensions are very important. Obviously, the same particle size, for example, in a smaller airway is going to behave differently than in a larger airway, and in particular, the branching pattern between rodents and humans is different. The rodent has a monopodial, which means that one of the branches is larger and longer, versus the primate system, which tends to be bimodal or bipodial, which means it's branching equally. The tissue dimensions and tissue volumes are very different, as well as the cell types and the distribution are different in each of the three regions, as well as the mucus composition. Each of these differences features in a conceptual model of dosimetry depending on the inhaled agent. Next slide.
Hey, Annie, this is Dave. Can I interrupt you for one moment? It seems that at least one participant is not able to see the slides. I can see them on my screen. Mariana, can you unmute yourself and can you see them? Okay, I'm hearing from others that there are no issues, so I'll continue to see if we can figure out what the problem is, but I'm getting multiple responses that folks can see the slide. So go ahead, Annie. Sorry for the interruption.
No worries. I'm sorry, I don't know why I wasn't able to advance them on my own. Likewise, comparative airway physiology is equally important and interacts with the anatomical considerations. Some principal concerns here are the ventilation rate and breathing mode. On the upper right-hand side, I'm showing an activity pattern for different types of activities, such as sleeping, sitting at your desk as a couch potato, or perhaps exercising. All of those activities are associated with a different exertion level and a ventilation rate to support that exertion. These are typically compiled into what is known as an activity pattern. For example, the National Institutes of Occupational Safety and Health and OSHA use a 8-hour per day assumption and different compilation of, for example, adult male light work is shown in the second row, and adult male heavy work, perhaps an hour of exercise in particular occupations. That can be used to characterize an occupational exposure. Likewise, the US EPA has a composite 20 meters cubed per day that we use as a default ventilation rate, but that's actually comprised of different amounts of activity for different periods of time. So if you have a model that can step you through that type of construct, you can actually vary the ventilation rate. Just appreciate that the default 20 meters cubed is a composite collapsed factor. The ventilation rate very much influences where particles and gases are taken up in the respiratory tract. We talk about proximal to distal distribution of both particles and gases, proximal in the upper airway, fractional penetration down to the distal pulmonary region. Also on the bottom right-hand side, I've got illustrated the switch in breathing mode. Breathing mode is also very important. As you increase your exertion level, oftentimes you will start to augment breathing through your nose with oral breathing. So that's a normal augmenter shown on the solid upper graph there. It shows the switching point. You up to a certain ventilation rate, you're fine just breathing through your nose. As you exert more, you have to switch and augment that breathing to oral nasal breathing. Some people are mouth breathers throughout the time. Those different types of breathing modes have very different deposition and uptake patterns in the respiratory tract. Also to consider in terms of physiological parameters are differences in mucociliary rates across species and the metabolic capacities of those cells that may be different in each region as well as distributed differently. Next slide. Physicochemical properties that interact then with the anatomy and physiology are shown here. Particles, fibers, and engineered nanomaterials. The principal ones are the density, the dimensions and distribution, how biodurable it is, typically based on its solubility. Some other factors there are listed. For gases, we need to address the molecular diffusivity of the gas as it comes in to the airway via convection. How is it going to diffuse to the airway wall? Whether it's going to react via hydrolysis or direct binding, perhaps metabolism in specific tissues, depending on the enzyme constituency of those tissues, or whether it's soluble. Perhaps it only goes directly into the pulmonary region and partitions into the blood, and the propensity there is more systemic toxicity. Those are the types of considerations that need to be looked at when you're considering the physicochemical properties and what part of the respiratory tract might be relevant. Next slide. Those same properties also interact with the exposure conditions and create some considerations for extrapolation. In general, for generation and characterization, you need to be aware of what the analytical methods and detection limits are. For example, the sizing convention for particles. On the right-hand side, briefly, is an illustration of the assumption that is imposed when you only characterize fibers with an aspect ratio of 3:1. The line on the upper left-hand side illustrates on the isoclast the amount of aerosol that you would be missing, and likewise on the inhaled dose on the right-hand side, the red illustrates the amount of aerosol that you would be missing when you use fiber aspect ratio such as 3:1 instead of using the bivariate distribution, the actual length and diameter co-characterized for a fiber particle. Concentration obviously is another important parameter. The duration of the in vivo exposure, be it hours, days, weeks, or years. The experimental regimen within that duration, such as 6 hours per day and 5 days per week. That influences whether or not steady state for a gas, for example, or a periodic exposure pattern was reached. And that is why time course data are very important to have when you're trying to characterize the dosimetry of inhaled particles and gases. So absorption, distribution, metabolism, and elimination, ADME, very much need to be characterized by a time course, as well as tissue reactions. On the right-hand side, I illustrate achievement of periodicity over time. That is that during a 6 hour per day, 5 day per week pattern, as you plateau, you get the same pattern achieved throughout the rest of the exposure. That is what we would typically like to have when we're extrapolating across different exposure conditions. If you don't have that, it takes a much more rigorous model to actually describe the excursions on a daily basis. On the far right-hand side are two parameters that help to tell you whether or not periodicity is likely to occur within a specific time frame, be it a chronic or subchronic exposure period. Finally, the other consideration for exposure conditions, particularly when extrapolating or trying to set context for interpreting in vitro as well as in vivo data, is the target human exposure scenario. You'd want to know whether or not you're trying to target an acute emergent situation or episodic, ambient versus occupational. I already mentioned, for example, that there might be different ventilatory activity patterns that you would want to assume to start to construct that exposure scenario. Next slide. So setting this in context then with contemporary toxicology, we very much have new bioassays coming on board and databases that are much more comprehensive in scope, particularly across different levels of observation, from genomic profiling up to biomonitoring in populations. Increased sophistication of measurements, we get to know much more what those cellular interactions and tissue reactions might be. This imparts a growing understanding of mechanisms at the molecular level. We can start to then construct models of pathogenesis in different disease states, start to understand susceptibility factors, and we now have enhanced computational capacity to describe those processes quantitatively. So how best to take advantage of these new technologies and understandings. Next slide. The way we have been doing that quantitative construct is with mechanistic modeling, which is a qualitative agreement with that biological understanding, appreciating that the biological understanding of the process is evolving. A quantitative agreement with the existing data that you might get. Again, that depends on your experimental design as well. Trying to validate the model through the prediction of the experimental data that was not used in your model construction. And then comparisons that are quantitative, looking at critical parameters characterized across species, and this is then consistent with contemporary toxicology construct that I just mentioned. Next slide. So in terms of risk assessment, what does dosimetry modeling mean, and how do we define it operationally to get to a risk assessment? So dose imparts that we are trying to describe the exposure versus we're trying to describe the internal amount versus the exposure. For an acute toxicity test, for example, it would be the particle deposited. If it was a chronic exposure, you'd be more interested in the retained dose. A metric for gases might be parent or metabolite. The definition of the dose metric is best described as at least the metric best associated or correlated with the toxicity or key event that you're trying to evaluate or extrapolate as the basis of your assessment. The metric can be whatever measurement you have, in which, again, is causal or at least best associated or correlated. So it might be particle mass or surface area, even number in some cases. For gases, it could be the blood or tissue concentration or the area under the curve or perhaps receptor binding. It depends on the mechanism. The scale of the metric for your modeling or extrapolation should be the same as the observation or response endpoint. So if you only have data in the lung region, that would motivate one metric versus a much more local specific cell type if you've got that type of data. And the model then is conceptual or the quantitative description of your understanding of the important processes and the understanding of the biology. And then you can then use the model to simulate the different exposure scenarios for extrapolation across the different experimental designs. So that, in a nutshell, is what dosimetry modeling, how it operates in risk assessment. Next slide. So there is a long history of dosimetry modeling. It actually started with radionuclides in the 40s. And the reason for that is that became a very clean system, if you will. They knew what the different particles, beta, gamma, alpha, how those disintegrated. They could measure it in the respiratory tract. The International Commission on Radiological Protection in 1994 published reference MAN, gave a lot of the dimensions of these different ventilation rates. That activity pattern that I showed you previously was from the International Commission on Radiological Protection. So that's really when it started, and it's evolved rapidly since then. Dosimetry modeling has a long precedent of being informative to risk assessment. For example, it's the basis for the size selective sampling used in the National Ambient Air Quality Standard effort for particulate matter. Dr. Gata mentioned 10 microns for humans. It's 2.5 in rats that's respirable, and you can fractionate those even further based on now our understanding of the differences in deposition in the different respiratory regions. So the aerodynamics of the physics of particle deposition is dependent on those factors that I just mentioned, the anatomy and the airflow, and primarily on the particle size, the distribution, and the density of your aerosol. The transport is dictated by those dimensions and the ventilation rate. Inhalability is very different. I've spoken to you about breathing mode. There's different ways to describe the fundamental first principles of the conservation of mass and momentum. You've got different algorithms for each of the types of deposition mechanisms. I'll show that in a moment. And for fibers, for example, you also have to take into account their orientation. Is the fiber entering parallel or perpendicular to the airflow? And that typically best requires the bivariate distribution for accurate characterization. Next slide. So here's a rendering of the respiratory tract model used for particles. Again, there's the three regions, the upper respiratory tract, the tracheobronchial, and the pulmonary region. And each region then is described, deposition in each region is described on a semi-empirical basis. And what that means is that you might have measurements that are made that actually sampled what the upper airway, the deposition in the upper airway was after a certain period of time. And that's also then coupled with these equations of fundamental first principles of physics for those specific mechanisms. So there's inhalability, impaction, diffusion, sedimentation, and those are all each described as separate algorithms that depend very much on species-specific architecture and then the airflow that can be developed in that architecture via different activity patterns. the different deposition mechanisms, if you look at the inset there, are very much due to the directional change that's occurring because of the differences in the airway architecture. So for example, in the nose, you've got much more impaction because the air velocity is quite high as they breathe, as an animal or a human breathes in the nose. Whereas diffusion is a much more dominant mechanism in the pulmonary region because the air exchange is much slower and at a low velocity at that point. So it's important to note that the relative contribution of each mechanism is different in each region of the respiratory tract. It's relative within the human, but it's also a different relative set of deposition mechanisms, obviously, in the rat respiratory tract. It's important to note the equation in purple, which is that the retained burden is the inhalability and the deposition minus clearance. So depending on what type of endpoint is your target, acute endpoint, a particular model might be good for acute endpoints with inhalability and deposition only. If you're targeting longer term 2-year bioassay data, then you would want to look at a clearance model for chronic exposures. This particular model is deposition only. There's a very similar construct that's used to describe clearance mechanisms in the respiratory tract amongst the 3 regions and then out to the interstitium. And the major deficit clearance mechanisms, would then be mucociliary transport, dissolution, and lymphatic transport. I don't have that illustrated. Next slide. So typically what we have used, and it has some precedent in that it's used for the particulate matter National Ambient Air Quality Standard science evaluations in the agency, as well as used in pharmaceutical delivery device calculations, is what is known as the multi-path particle dosimetry model. It is based to some degree on the International Commission on Radiological Protection, but it has been established in regulatory practice. It does have updated algorithms and enhanced deposition efficiencies utilized, so that's why we choose it for the model. And both EPA and NIOSH use this. We are currently working on extending it to some fiber descriptions, and NIOSH has been working with it to describe nanoparticles. Next slide. Just want to highlight for the in vitro, in vivo extrapolation that those same mechanisms that I just described in terms of transport and transformation processes, diffusion, sedimentation, a mixture of them, are also very much relevant to the in vitro situation. So there has been a model developed by Paul Hinderleder and others, the computational model of particle sedimentation, diffusion, and target cell dosimetry for in vitro toxicity studies. They have a very elegant presentation in their manuscript that shows that you must consider these very same transport mechanisms to better describe and characterize delivered dose to the cell. And for the purposes of risk assessment, you would then want to try to run simulations with this model to compare with what your target scenario might be, occupational or ambient, for example, using that in the multi-path particle dosimetry model to extrapolate the doses. Next slide. Just to illustrate what a difference the dose does make, this is the recent work I've been doing with fibers in the multi-path particle dosimetry model to illustrate the difference between looking at the number of fibers deposited in different regions of the respiratory tract. Each of the regions is represented in a different color. The upper airway in blue, tracheobronchial in red, pulmonary in green, or the composite lower respiratory tract in the yellow. There's humans on the upper at a particle aspect ratio of 3 versus humans in the lower panels with a particle aspect ratio, fiber aspect ratio, of 10. Number of fibers is illustrated on the left-hand side. Surface area is located on the right-hand side panel. So you can see a dramatically different pattern of internal dose depending on how what you're characterizing in terms of the size of the aerosol coming in, as well as whether you're choosing to represent the metric as a number versus surface area. This is very important when you're trying to best match the metric to what might be the mode of action. So the metric and the aspect ratio determine the magnitude, which region it actually deposits in, and to what degree. Species differences aren't shown, but you can imagine that the differences in architecture features prominently. Next slide. In terms of gas uptake, the conceptual construct is shown in this slide. You've got convection in the airway on the left-hand side. The comparative features that determine that airflow is the ventilation rate, the geometry, the airflow diffusivity, and then once the molecule of the gas hits the airway, other features that factor in to describe its absorption is the diffusion in the tissue, the solubility, its metabolism rates, and what reactions there might be, as well as the tissue dimensions. It's going to be different in the rat versus the human. And then likewise, once it gets to the blood flow, what the partition coefficient of the physicochemical gas might be, whether it's more soluble or less soluble, as well as reaction rates in blood constituents. So a typical compartmental model describes those functions. Shown on the left-hand side is a compartmental model. You've got the airflow describing the convection. Superimposed on that is the molecular diffusion to the airway wall, and then you have a concentration gradient from the middle of the lumen, for example, going to the airway wall, and then subsequent decrease of the concentration of that chemical through the tissue, be it mucus, the epithelial layers. You've got continued metabolism and diffusion and into the blood flow. So the left-hand side is the airway factors in the blue, and then tissue on the right-hand side. And it also depends on whether or not, when you're creating that dose metric in the tissue, for example, are you interested in the parent or the metabolite? One example of what a difference the dose makes is on the right-hand side. This is work done with Nat Bogdancy on vinyl acetate uptake. The fractional deposition or uptake of vinyl acetate is on the y-axis versus exposure concentration. If you made no adjustment for metabolism, you would get the blue diamonds. If you did consider tissue carboxyl esterase to some degree with low affinity, you get the green. The actual observed data is the red, so clearly neither of those two first dose metrics describe the data well. You needed to include the distribution of a high affinity carboxyl esterase in the mucus to best describe, as shown in the purple line, the uptake of the gas. This becomes very important because the mode of action for vinyl acetate was then uptake metabolism to acetaldehyde, potentially related to the tumor formation as well as cytotoxicity. I don't have time to explain the full biologically based dose response model that we developed there, but illustrating that these parameters are very important to a more accurate description of gas uptake, just like it was for particles. Next slide. So a generalized structure for gases then is shown here. You've got the extrathoracic region or upper airway on the left-hand side, the tracheobronchial and the pulmonary region. You can either represent those compartments as a fractional penetration going one from the next, or you might just isolate the one region and describe your dose metric within that. The sequential regions do allow you to account for fractional penetration. Most models do not address the exhalation, the CX exhalation coming back on breath. Only inhalation is typically used. The tissue compartments can be the regional, as I've illustrated, or more localized, which I'll illustrate on the next slide. And the blood flow links to remote compartments should you want to describe systemic toxicity from an inhalation exposure. Oftentimes, we will simplify and just say, for example, if the toxicity is in the upper airway, you only use a one compartment model to describe the upper airway, and there may be just some default algorithms additionally to make it even simpler. In some cases, a more complex model such as this is needed. So you can either have a single path mass transfer type of construct, looking at mass through these regions if it fractionally penetrates proximal to distal, upper airway to the pulmonary, or perhaps a sequential set of compartmental models are often used to describe this type of uptake in the airway. Next slide. For more localized and sophisticated modeling and really trying to understand better a pathogenesis process, again, I hope I've illustrated that we need to address those differences in dosimetry in a comparative fashion between species with respect to anatomy, physiology, physicochemical properties, and exposure conditions. Illustrated here are the nose diagrams and differences in airflow in the human in the upper and laboratory rat in the lower. Would note that the nose has all the epithelial types that are found in the lower respiratory tract, so oftentimes we feel that the nose is a model for the lower respiratory tract. It has respiratory epithelium, olfactory epithelium, and ciliated epithelium. It's very important to know where the gas distributes and as well as the distribution of the cell types to do accurate accounting of what the metabolism, particularly for upper airway lesions, might be. Models have matured and are rapidly becoming more mechanistic to address this type of localized construct. Would also note that in the lower airway, these models are now being developed for the lower airway. What I've got illustrated at the lower bottom is Rick Corley's very elegant work looking at computational fluid dynamic models of the nasal airway, again now being extended to the lower airway, and being able to understand the pathogenesis, not being restricted to any particular testing paradigm, but then to now actually be able to model and simulate exposure conditions to estimate different dose metrics. So on the left-hand side is surface flux. The red illustrates a high flux area. The blue is a lower concentration. Clearly a very different pattern between surface flux as the dose metric versus area under the tissue, area under the curve for tissue concentrations. That would be a big influence on what you then derived as your point of departure and basis for a risk assessment depending on your metric. So you can, what I've tried to illustrate here is the breadth of types of modeling being used and what a difference it does make in terms of what your metric might be and to what degree you are characterizing it with a model or a default assumption. Next slide. So to create that context for application in risk assessment, the human equivalent concentration is constructed, and that equation is shown on this slide. The point of departure can be, for example, a no observed adverse effect level, or more typically a benchmark dose level fit to dose response data of your particular endpoint of concern. So the point of departure adjusted for the exposure regimen. So if you're adjusting to an in vivo situation in a rat, it would be the number of hours per day times the number of days per week the rat was exposed times a dosimetric adjustment factor. And that dosimetric adjustment factor is different. It considers those conceptual models I've just reviewed, be it particle or gas, to create a ratio between the animal and human to adjust then to a human equivalent concentration. Work is underway as those models mature to replace both the right-hand side with dosimetry modeling. For example, the dosimetry models that account for metabolism, you don't need to adjust for that exposure regimen because that is explicitly characterized in the model simulation. On the left-hand side, you can replace it with response modeling in terms of what you believe the adverse outcome pathway is and the tissue responses. But that's your fundamental construct, and then you can try to improve both sides. Next slide. On this slide, I illustrate that calculation for particles. The regional deposited dose ratio is based on the deposited dose of particles in the animal versus the human in the region of interest. So for example, if it was in the pulmonary region, you would be using the fraction deposited predicted with the model for the pulmonary region, and you would be adjusting for normalizing factors relevant to the pulmonary region, such as you wanted to normalize to the surface area or perhaps to a dominant cell type such as the alveolar macrophage. You adjust for the minute volume or that activity pattern with the ventilation rate, and you can create that ratio to then calculate the human equivalent concentration. Next slide. This extrapolation then is illustrated schematically on this slide to show how you can relate the tissue responses either in vivo across species. It's also immediately applicable and analogous for in vitro situations. So you start with your animal or in vitro exposure, you account for the dosimetry in that particular species or test condition, you estimate your internal tissue dose, and then you look at what your response is. You might have a model to also characterize the response components. You extrapolate based on that tissue response using the same considerations and components, response components and dosimetry components, to a human equivalent exposure. Those human equivalent exposures, be it across species or across in vitro to in vivo, would be what you would use as your comparison framework. So you'd be expressing the different exposures based on a human equivalent exposure. Next slide. So bridging then to systems biology is in that conceptual computational approach is shown on this slide just to kind of capture and summarize. You construct your conceptual model based on your current understanding of the biology and the relevant parameters. You define and measure those parameters and implement a computational model, and then you can evaluate in the comparative context. So in this case, a full-blown biologically based dose response model could be a computational fluid dynamics model, telling you what the deposition or flux to the tissue was, either in the region or a localized epithelial type. You might have a physiologically based pharmacokinetic model to describe the disposition within the tissue, such as tissue reactions, particular enzymes, such as shown with the carboxyl esterase and vinyl acetate, and then evaluate the response endpoints. There might be additional tissue responses that you need to characterize with the model. That same sort of construct, it can be used in the in vitro situation, again, and then compare with in vivo, be it laboratory animal or human. Next slide. Model selection criteria are shown here. Obviously, the availability of the data and your ability to go get the data that you need to define the parameters, physicochemical characteristics, first cut particle or gas, the location of the toxicity, is it in the upper airway or is it in the pulmonary region, and what level do you need the description, cellular or population, toxicity time frame, the mode of action, and the metric description, as I've already reviewed. Next slide. So there is a hierarchy then of the model structure, and that influences what your degree of confidence in the characterization or the resultant uncertainty might be. At the low end, you might have just the exposure adjustment. Perhaps default toxicokinetic differences in animals and humans can be applied. Categorical defaults include modeling such as allometric scaling, looking at the differences based on body weight scaling or differences in ventilation rate. What I've shown illustrated is the generalized construct, and just looking at the upper airway, there are reduced form analytical solutions that just looks at what is the uptake in one architecture versus another for the upper airway. And this is typically coupled with data-derived uncertainty factors if you have toxicokinetic data that might support that. Getting to more sophisticated, you've got the computational fluid dynamics, that of Corley that I illustrated there, or single pass mass transfer, looking at just what the uptake might be in each region. If you need to have better descriptions of the tissue distribution and the physiology, perhaps within one specific type of epithelium, you can move to the physiologically based pharmacokinetic model. Again, data-derived uncertainty factors, or preferably that comprehensive biologically based dose response model. That sort of is the gamut of the type of modeling approaches that are available, and I wanted to give a sense to you of what was available so that you can create that context for where do your in vitro data fit relative to addressing some of these biological, anatomical, physiological, and physicochemical properties. Next slide. So moving on to a larger context for risk assessment, we're really moving towards multi-scale modeling. The type of approach and modeling that we're trying to do needs to be flexible to be able to characterize the different exposure scenarios. We're moving to address different scales, be those temporal, acute versus chronic, biological, cellular, or tissue, population, geographical, and then to describe the disease dimension. Is the particular endpoint or marker that you're using, does that represent, or is it a surrogate for an early or late event with respect to the key events of a pathogenesis as a disease develops, and to try to develop probabilistic approaches of those key events, which would help then cost-benefit analysis as well. So that's the large-scale view of where we're going with risk assessment. Next slide. And that type of modeling really needs to be done, as I've illustrated with dosimetry models, to provide the translation of dose and data integration across these different levels. So this is a very beautiful slide from Bill Benson where it shows at the community level what we might want to be characterizing all the way down to high throughput, high content testing, genomic profiling, clinical chemistry, biomonitoring. We need something that is flexible and contextual so that we can integrate data across all these various levels. Next slide. So one construct that's been offered and is advancing is the adverse outcome pathway and mode of action in risk assessment to provide that data integration construct. So illustrated here is the biomarker scheme that was used by the NAS in the late 90s, and onto that I have superimposed what we use as a mode of action model, starting at exposure on the left-hand side, moving down through exposure to the internal dose, and then onto particular adverse outcomes relevant to human risk assessment. In the blue is where the adverse outcome pathway construct fits, starting with the molecular initiating event, as Dr. Gada illustrated, to an adverse outcome. This entire schema fits together, and what we really need to be able to do with these dosimetry models is to characterize the exposure factors that take you from external exposure to an internal dose, and then defining the dose down to the molecular initiating event. So there's different levels of organization, tissue to cell receptor, for example, and then describe with dosimetry and response modeling, again, that full-blown biologically based dose response construct, the subsequent process of pathogenesis for a particular disease. All along the way, there are factors shown in the ovals that are dashed intersecting the response, the key event relationships that are both dependent on concentration and duration of exposure. So a full characterization requires understanding a pathway and what those dosimetry concerns are, be it the temporal duration of exposure or the physicochemical properties interacting with the biology, to construct the sequence. We do feel that this is possible, particularly for portal of entry descriptions. It's actually requisite to really be able to understand what's happening in the respiratory tract, and eventually we will be able to do this in a broad context for several endpoints and chemical classes within the respiratory tract. Next slide. So creating that context from in vivo to in vitro extrapolation is shown in this slide from Bas Blaubar. He talks about how you evaluate risk, obviously the exposure assessment, as I've mentioned, looking at the physicochemical properties, understanding the ADME, the biokinetic properties, determining the appropriate tests and conditions, and then performing your battery reconsideration and iteration in that modeling construct of those dosimetric as well as response components to characterize the exposure dose response continuum and conceptual model, as I've already discussed. Next slide. So in terms of aiding your experimental design, you really need to think about these considerations advance, particularly creating the context for the comparisons. I've got illustrated on the right-hand side epidemiological characterization of an ambient aerosol. On the very far right is what the PM10 sample would show you in terms of the resultant aerosol characterization. That's very different than what's actually being deposited in the thoracic cavity of the respiratory tract. So you've got to look at what is the sample method and its analytical limitations might be versus what's in the thoracic or whatever region of interest that you're trying to target. For in vivo studies, another consideration might be whether you've got an inhalation or an instilled type of exposure. And then in vitro, obviously, it's going to be different. Those transport and transformation mechanisms are going to be different if it's applied via the media or via the air. You're trying to estimate down to the cell level. This has an impact on the inferences. Either biases are introduced based on these that I've mentioned, and oftentimes the poor correlation is due to a failure to account for these determinants of dose and the causative events of response. So that's something to keep in mind. Next slide. So selecting the relevant dose metric for IVE comes back full circle to those major determinants in the conceptual model for the biology and the physicochemistry of inhaled dose. You want to have your selection of the mechanism and based on your hypothesized mode of action, so correspond it to the key event. Is your key event just a regional description, or is it actually cytotoxicity in a specific cell type, for example? And the temporality of the disease dimension, as we've mentioned, is it just acute toxicity testing and acute endpoints, or are you trying to relate your acute testing assay to chronic endpoints? You need to account for the key characteristics of exposure, those individual anatomical and physiological parameters, particularly age-specific anatomy and ventilation rates, the cell types, the breathing mode, the activity pattern, and then, of course, the major physicochemical properties that I've mentioned with respect to particles and gases. Next slide. Finally, the advantages to this type of construct of mechanistic modeling is that it does represent the current and best understanding today of the biology and physicochemical mechanisms for a given mode of action. It aids comparison and translation of those results across exposure conditions and between species. You can target specific scenarios, and it facilitates comparisons depending on your dose metrics with the disease endpoints that you have either measured or want to provide insight on with respect to mode of action. And ultimately, this does refine risk assessment predictions. Next slide. Thank you for your attention and look forward to discussion.
Okay, thanks very much, Amy and Mariana, both. Apologies that we have run a little long, but I think everyone will agree. It's been a really great overview and discussion of the available toolbox associated with alternatives for inhalation tox testing and the important considerations in modeling exposure and dosimetry associated with inhalation tox. Given the late time, we do have a few questions that have come in, but what we'll do is we'll have an electronic response to those after we disconnect today. And please, if others have questions, Amy, the screen right now has our contact email addresses. Please do email either Amy or myself with any additional questions, and we'll be sure to follow up with you in short order. So thanks again, everyone, and have a great afternoon.